Welcome to Max Planck Florida Institute for Neuroscience's Neurotransmissions podcast. I'm Jeremy Chang, and today I'm joined by Akash Pal.
Heyo.
So Akash, today we got the chance to speak with Dr. Tony Movshon. He's a professor of neuroscience at NYU in the Center for Neurosciences, where his lab studies how the brain encodes and decodes information about the world. He's also a repeat guest, having been on a podcast previously. He gave this really compelling argument against why connectomics may not be the way to approach questions in neuroscience.
Today we did get an opportunity to talk more about his science relating to the visual field. We are also going to talk about a reductionist and a constructionist approach to science, and he's going to tell us a lot of things about the visual pathway, how it works. I think it's going to be a great podcast.
Yep, and as usual, Dr. Movshon always leaves little tidbits for aspiring scientists, and
He does.
So I guess without any further ado, we'll throw to our conversation with Dr. Movshon. So Dr. Movshon, your lab has been focusing on multiple aspects of how visual cortex encodes information about the visual world. What is it that actually got you into studying visual perception to begin with?
So I think what got me into studying vision is what probably, if people admit it, got most people into the things they study, which is somebody who was interesting to them was working on that problem, and they got interested in the problem that was interesting to that person. So in my case, the person who became my PhD advisor, Colin Blakemore, was one of my teachers as an undergraduate. And I was interested in his research because he was a very compelling exponent of why it was interesting. And why. It also happens that where I was at the time, which was in Cambridge, was a sort of a hotbed of work on vision and visual systems. So there were lots of people to talk to and lots of people to get interesting aspects for. So I did not grow up saying I wanted to be a vision scientist. I grew up, probably saying I wanted to be a fireman like everybody else. But I sort of fell into vision because of the people I ran into.
One of the interesting things is your approach to actually studying the visual system, right? I think a lot of us have been classically trained as reductionists, thinking at things at like the individual at the simplest scale as possible. But you've sort of taken a different approach to how you've studied vision. Could you tell us a little bit about what that approach is and how it maybe changes the questions you're asking?
Right. So, I mean, I think I was inspired and continue to be inspired by people like Horace Barlow and David Marr and others who have thought about the problems of vision, how you actually handle the question of interpreting the visual world and turning the sort of photoreceptor signals that reach your eye into a meaningful, interpreted representation of what's in front of you. It's common to think about vision or any sensory system as though it were a physical measuring device. Right? This is a device for measuring light or for measuring sound or for measuring pressure on the skin. And senses are none of those things. Senses are devices for learning about the world, and especially senses like vision, which are for remote sensing, are about really inferring what things in the world give rise to the pattern of photons that reaches the retina. And that's not a sensation problem, that's a computational problem. It's a problem of inference. People who started studying that and thinking about it, many people over the centuries have thought about it, but Helmholtz was particularly important in that thinking. And then there were great psychologists like Richard Gregory, who basically posed the problem as an inference problem, which is the visual system, you should think of it as being like a scientist. It's faced with a set of data. And on that set of data, it has to impose an interpretation, which is the interpretation of what factors in the world give rise to that set of data. That, in the case of a scientist, is a research program. In the case of the visual system, it's a percept. What we see is what we believe to be true about the world that gave rise to what reaches the eye. What reaches the eye is, on the one hand, it's a very rich signal because there are millions of photons and photoreceptor activations and so on and so forth. On the other hand, it's a very impoverished signal because it doesn't carry any information about anything in the world that you care about, where this cup sits, where this microphone sits, whether this is a table or not. And all of those things have to be deduced and computed. And so people like Gregory, Helmholtz, and then also people like Barlow and David Marr, both of whom I mentioned today when I spoke, really thought very carefully about what we should say about how the visual system is designed, how it encodes information in a way that is most efficient for extracting this sort of knowledge. And the things that I've tried to do are really to put neurobiological clothes on that intellectual skeleton. So suppose that metaphor isn't particularly appealing, but because you probably want some muscle and skin under there as well. But that's the idea. The idea is to try and understand in terms of biology how the visual computations that underlie things that seem effortless to us, like straightforward percepts, are actually done in the brain.
That's actually a nice segue, because what I wanted to ask you was that, nowadays with our experience with computers and digital cameras, people think of vision as being like a single pixel that has all the brightness and the color information in it. Obviously, that's not how the visual system encodes. So can you tell us a little bit about can you explain a little bit about some aspects of visual scenes and how the visual cortex encodes these?
Yeah, well, so it's easier to say things about scenes than it is to say how the visual cortex encodes them. But so, for example, vision begins with photoreceptors, and photoreceptors are like the pixels in a digital camera. They encode, local patterns of light intensity, and in a limited sort of way, the local wavelengths. So, you have, in daylight vision, you have 3 photoreceptor types that give you information about different but overlapping bands of wavelengths, so you can infer the spectral content of images from those. But knowing the spectral content of an image is only the beginning. So if you're looking at the world, if you look at the whatever her name is, who's the goddess? It's Athena, Minerva. Minerva's the Roman one, isn't she? Athena? No. Who's Minerva in Greek? Minerva's the Roman name. Anyway, Minerva. We'll go with Minerva. The pattern that gives rise to the image of Minerva, could, in the simplest form, consist of either variations in the surface texture of the object on which she's painted, or it could be created by a projected pattern of light. Right? What's the difference between light and paint is a very simple question about vision. How do you tell the difference between a pattern that emerges from the surface properties, and how do you distinguish that from a pattern that emerges from illumination? So there's a shadow at the edge of that disk with Minerva's head on it, and I know immediately that that's a shadow cast by the overlying light and not a feature of the tablecloth underneath. Mm-hmm. Solving problems like that, sort of very elementary, but since the visual system, or it seems very, very elementary, it's not actually computationally elementary at all. And one of the things that's sort of interesting is that it's something that the visual system has to develop. There's a bit of a digression, but my colleague Pavan Sinha at MIT has done a lot of work with Indian patients who had cataracts, which were removed when they were adults. So these were people, normally with Western medicine, they would be treated early and they would have had clear vision early in life. But for whatever reason, so he started this thing called Project Prakash, which takes basically mobile medical treatment throughout India. And one of the things they do a lot of is removing cataracts. And what they do is they create a group of patients who can see for the first time clearly after many years of occluded vision created by the cataract. And one of the things that they have to learn is the difference between shadows and surface features and lights, and how these are all disambiguated in the visual world. So their low-level vision is quite good. They can see fine detail pretty well. They can see things reasonably easily in terms of their overall levels of contrast. But perceptual interpretation, scene organization, is something they do very badly. And they learn how to do that later. So that's the difference between knowing that thing is a shadow on the tablecloth and not a dark patch on the tablecloth.
Right. So there's some sort of, previous experience that is informing your interpretation of the actual visual, what the photons are doing when they hit your eye.
Exactly. There's a process of learning, developmental process of learning. Now, I have done work on visual development, and there's work going on here about visual development, which is focused on more low-level questions like how the two eyes get wired up together and things of that sort. This is a higher-level problem. Mm-hmm. It's not one we understand so well. But for example, there's a simple version of this which I kind of like to teach. It's not my own work. It's the work of von der Heijden and his colleagues at Hopkins, and it focuses on something that's called border ownership, which is a very simple concept that basically says if you see an edge in a scene, the edge that this coffee cup creates in the scene, it's created by an object which is occluding a background. And therefore, you can say, in the sense of foreground and background, that this object in the foreground owns the edge. It owns the border. And so the concept of border ownership is that this border here that I can see in front of the table is created by the occluding object and therefore belongs to this object. It turns out that cells quite early in the visual pathway are sensitive to border ownership. They can actually tell the difference between a case like this, where the border is owned by this object, and a case which is locally identical, where you have a dark contour on a light background, but there's no border ownership signal. And so that computation can be done quite early in the visual pathway. Exactly how it's done is a computational question, which various people have tried to explore by looking for the image features that create it. That's one example of the kind of problem that I think is an interesting step from the simple encoding of an edge, which we sort of know how that gets done, to the interpretation of the edge. Is that edge created because there is actually an occluding object which creates the border, or is it an edge which is created by some other thing, like a surface feature on a tablecloth?
And so your lab has really been focusing on sort of this, I guess, the construction of these basic features into more complex aspects of visual scenes, and you've been looking particularly at higher cortical areas. Could you tell us a little bit about how things sort of are being constructed as you go further down the visual pathway?
Sure, I can give you a couple of examples. So we've worked on a couple of different versions of this, and what I would say is where we work is less high level than mid level. So there's a sort of a loose concept of mid level vision that we use and a lot of other people sort of use to describe something that falls short of the sort of level of object analysis that you see in the far temporal lobe reaches of the visual pathway. But so, for example, let's take a case of visual motion, which is something I've worked on quite a bit in the past. I didn't talk about it today. But what we did many years ago was to establish that there seemed to be multiple stages in the analysis of visual motion signals. They, again, sort of have the same notion of at the beginning of a border. So if you create a border and you put it in motion, it passes across the retina, and you have a basic simple problem of encoding that information. And you can do that, and the first cells in the visual pathway that are motion sensitive or sensitive to motion direction, which are in the early parts of primary visual cortex, signal the direction of motion perfectly well. But the question of how you interpret that motion is different, because that moving border often belongs to an object which has a complicated shape. And if you have an object with a complicated shape, its borders form many different contours. And each of those contours, if you analyze them separately, has a different movement. This is something which perceptual psychologists have known for a long time. They call it the aperture problem. Because if you look at a moving object through a small aperture, the object moves past, you can only see the motion locally of a piece of the object. But when you open out the aperture so you can see the whole shape, you realize that this part of the object moves like so, and another part of the object moves in a different direction. And the job of combining those signals turns out to take place downstream from primary visual cortex, and it probably starts to take place in an area called MT, which is the sort of second stage visual area, which combines signals from cells in primary visual cortex. And cells in that area give responses that are invariant with the true motion of objects in 2 dimensions. Further downstream still, we think there are other cells that are sensitive to the motion of objects in depth, for example, to the deformations of objects, which you can infer from their motions and so on. And so the way we tend to think about this is as a sort of cascaded series of computations, each relatively simple, sort of transforming, for example, the computation that takes non-direction selective signals in primary visual cortex and computes direction selectivity is something that we, at least in principle, understand fairly well. The computation by which we take these local, what we call 1D motion signals, and combine them to make 2D motion representations is also something we understand conceptually fairly well. Now, somebody who wanted to know the exact circuit basis of that would be disappointed, because we don't know that at this stage. Mm-hmm. But the overall computation and its representation in the responses of groups of neurons is now pretty well documented. And so it's that process for me of taking first stage signals that we think we understand and going down to the next stage. So today I talked about work involving processing of information about texture and shape. Same kind of approach applies there, and we sort of think about those processes as having meant much of the same computational architecture as the problems in motion.
So from today's talk, when you were showing us the difference between, the actual image and the synthetic image that you generate through this algorithm.
They were both actual images. It's just one was made up.
Yes. So one was made up. Can you tell us a little bit more about this algorithm that you used to?
Sure. I mean, that was not my work. That's the work of Javier Portilla and Eero Simoncelli. And the way that algorithm works is it takes an image of anything, but it's convenient if the image is a reasonably localized image of something that is self-similar. Maybe we can at some stage paste on the image of Feynman's face, which is of course a case where the texture model really doesn't work very well. But the way the texture model works is that it simulates the output of a patch of V1, right, containing a bunch of cells sensitive to the local orientation and size of the elements of the image. And so it takes that sample, basically makes a list of all the responses of those cells. And then it takes all of those responses and asks how correlated they are across the image. In other words, if, for example, you were looking at an image of just white noise speckle, with no structure, then the correlation would be expected to be zero. Any correlations you would measure between sensors in the image would be accidental. But in the face of natural images, when you look at real images with real structure, there is a regularity across the image, and that creates a regularity in the correlation pattern across those initial V1, we'll call them sensors. We call them receptive fields or sensors, whatever you want. Depends if you're doing computer vision talk or brain talk, but let's call them sensors. Now, I can make a list of the sensor activations, and I can make a list of their correlations, right? So then I can throw away the original image, and I can take a piece of white noise, and this is where, what Javier and Eero did is where the magic works. I can put it in the computer, and I can basically push it so that its list of sensor activations and sensor correlations matches the ones I measured from the original ancestral image. And I can keep pushing it by an iterative process until I get an acceptable match, at which point I have created a synthetic texture which is based on the statistics of the original image, but is not the original image. And I can do that any number of times, just starting with a different piece of white noise to start with. And what I can then do is ask how those correlational features make a difference both to the perceptual appearance of the image, and people are actually quite good at discriminating different levels of that naturalistic texture signal, and what a difference it makes to neurons. And of course we discovered that neurons in V2 but not in V1 were sensitive to those texture components. It's quite analogous to the MT motion problem, because what happens in MT is that cells in MT that we call pattern motion sensitive cells combine signals from inputs from V1, and those are basically signals correlated by the motion of coherent objects. A coherently moving object like a texture with many features in it activates many of those cells together and causes a coherent pattern of activation. And the same is true of a pattern, a static image that forms in V1, or not a static image that forms in V1, but a static image activates V1 and creates that coherent pattern, which then activates cells in V2. V2 and MT are both one synapse downstream of V1, and maybe the calculation, actually we think that the calculation that V2 does and the calculation that MT does are conceptually similar, and maybe similar even at the synaptic level.
And so, kind of reading between the lines here, is the idea sort of that V2 and MT are sort of taking the basic statistics and trying to pull meaning out of them? Because it seems like these textures, you said they share a lot of common features, but one of them has an actual object associated with it, whereas the other is sort of this something that's more akin to noise.
Yeah, meaning is tough, okay?
Yeah.
So you can say one is, a first-order approximation to the original image. It only contains the same orientations and spatial frequencies. Then when you add the texture correlations, you get a second-order approximation to the original image. But you still haven't approximated the original image. Remember that the Feynman texture doesn't really look at all like Feynman's face. And that's where we wanted to go, and that's where the last part of the talk was about, which was going downstream to V4 and asking, could we find a transformation between the naturalistic textures and the actual original photographic images? You remember at the top of the pyramid there's a single photographic image, and from that we can go down through the scrambled series of textures, and from there we can go down to the noise texture. There's a whole series there which we can explore. And the idea is, and this I will admit is not as well formed as the earlier one, that the transformation that takes place between V2 and V4 is the same one that takes place between V1 and MT or between V1 and V2. That is, it's the same sort of relatively simple recombination of signals from the earlier area to the later area. That is, I should say, something between a hypothesis and a guess. And I can't document it for you other than by saying I can write down a model that sort of works for all of those processes.
So I'm curious, you've studied quite a bit in terms of the progression throughout the visual pathway and how this basic low-level properties of visual scenes are being composed into to actual coherent visual scenes. Where do you think visual neuroscience is headed, and what are the big questions that still remain to be answered?
Well, the big questions remaining to be answered are practically all of them, because I don't think we have good answers for almost all the interesting questions, but that's a sort of cheap answer. Where is visual neuroscience headed? So I think a big question that faces a lot of us in visual neuroscience at the moment is, what is the relationship between visual neuroscience, and in particular understanding visual cortical organization, and the properties of convolutional neural networks, which were, of course, as I mentioned today, in their original design, they were inspired by the architecture of the visual pathway. So the first convolutional networks were designed. I mean, the first one really, you can go back a long way to Fukushima had a fully working model in 1980 of sort of visual organization based on what he called perceptrons, and the perceptrons even go back further to a man called Rosenblatt who basically did there's some wonderful stuff from the late 50s based on truly ancient technology, which is again based on the idea that these simple combinatorial networks that combine low-level inputs into successive recombinations at higher and higher levels could be extremely powerful.
These are sort of quasi neurons, right?
Quasi neurons,
taking inputs, summing them, and then
So convolutional neural networks have a bunch of neuron-like features in them. They typically have a rectified linear output function. They do a pooling through a linear filter. They often do a sort of a max operation over a region of space. Sometimes they have things like gain controls built into them and so on. Convolutional networks were more a curiosity than a real model for a long time, because it turned out that we really didn't have the computational power to exploit them fully. And then there was a real breakthrough, which was led by Jeff Hinton, I think, probably principally among them, who was a computer scientist working in London and in Canada. What Hinton realized is that with the addition of a few tricks and tweaks and a lot more computational power, you could actually solve the problem of optimizing these networks. Mm-hmm. These networks are typically trained by a process called backpropagation of error, aka backprop. Not a biological process, not a process that people think of as, but training the networks with backprop and making backprop work was something that Hinton succeeded in doing, and he really succeeded in doing it 12 years ago. With the creation of something called AlexNet. Khrushchevsky was the first author, Alex. And so after that, there was just a huge cascade of successes for these deep convolutional networks. You can call them neural networks or not if you want. And there's been on the one hand, there's this huge success in terms of AI, which is really not my domain, and AI can do extraordinary things now using the power of these networks. And then there's this other question about whether models based on those networks are good models of vision, right? And the deep net models have been applied in a whole variety of different ways to different aspects of visual function. And although I'm an admirer of the visual system, and I'm an admirer of what convolutional neural networks can do, I'm not myself convinced at this stage that these models form good models for the visual system itself. I think that there's very good evidence that those models use computational strategies, which in some ways are very similar to the computational strategies that the visual system uses. Some people, I think of Jim DiCarlo and Dan Yammans, have argued very specifically that different levels of the models correspond to different levels of the visual pathway. And it is natural to think about them that way because they are based on cascaded computations that are sort of neuron-like. On the other hand, I think there are many aspects of the way those models are built and trained which are very different from the way that the visual system works. And so for me, the jury is out on whether those models represent good models of the visual pathway. And I guess my main hesitation about reaching into that space too far is I don't know what it means to say that I understand the visual pathway based on such a model, because these models are hugely complicated internally. They have uncountable numbers, well, countable but very large numbers of free parameters inside them, and you really don't know how to interpret them in terms of underlying biological mechanisms. So although I was and remain very much an articulate, very much an advocate of understanding systems in terms of their computational architecture and worrying less about their implementation, I think if you're making a model of the brain, you have to respect something about the architecture of the brain in order to leverage the understanding. That said, next year somebody could do something that convinced me otherwise. Anyway, so that's one set of answers about where the visual system is going, one I think has been dominating a lot of thinking, and a lot of people, a lot of the most active work in the part of the brain that I work in most, which is the extrastriate visual cortex of the non-human primate, has been driven in recent years by these deep net models and by the convolutional neural network models. Otherwise, I think what's happening in vision is kind of what's happening in most of systems neuroscience. We're concentrating more on questions of how populations of neurons respond. We're taking advantage of new technologies for making parallel recordings and understanding representations at scale over very large ensembles of neurons. We're relating the activity of those populations much more directly to behavior and behavioral circumstances. All of those things are powerful and important, but none of them is really unique to vision. I think vision has a particularly close relationship with the deep net models, and I think that's why I gave that part of the answer first.
Mm-hmm. So, you have been a PI for a long time. You're doing research for
Thank
a long
you.
time.
I'm old, yes, I agree.
So, what do you enjoy most about being a scientist, and what keeps you motivated and passionate?
Sometimes I wonder. Discovery is itself the reward. Discovery is just a kick. It's just a lot of fun to have something new come in the door, whether it comes in because, somebody sends me a figure from an experiment they've done, or somebody reinterprets some data using a new theoretical approach, or somebody comes through and proposes a new idea for how we can do a different experiment and set a different goal. I think all of these are exciting to somebody who cares about discovery, and I think all of us who are in this business are in it because we care about discovery. I'm not in it for the money. I'm not in it for the fame. I'm not in it for the gratification of being a senior faculty member at a major research institution. All of those things are fine. There are certainly worse ways to spend your life, but in this business, if you don't actually get excited by discovery, then you don't belong in this business. You should be somewhere else.
I mean, yeah, of course. And, new, fresh data is always exciting, but not all data.
No, not new, fresh data is always exciting. Some new, fresh data is just old, stale data recast.
So how do you deal with when, you have failures for experiments, when you don't get that data that gets you excited?
Failures are successes. No experiment is a failure. An unexpected outcome is what we should all be hoping for in science, because what we should all be trying to do is to find the points at which our ideas fail, to find the points at which we actually don't succeed in predicting the outcome. If we spend our entire careers predicting outcomes and then getting successful answers to those experiments, then we're clearly doing something not very adventurous and not very interesting. And I'll go back to this business about cells selective for the motion of visual patterns. I and a couple of postdocs, really just one postdoc, predicted that these cells should exist on the basis of sort of psychological and theoretical considerations. And so we started looking for them with electrode recordings in the brains, first of cats and subsequently of monkeys. And we kept failing to find them. So those were successive failures. I was sufficiently convinced that this stuff was going to work that I would even give talks reporting the failures, even though I knew that there were no successes yet in the can, because I was quite sure the perceptual effects were very interesting and very vivid, and I was quite sure that we would find, in the end, somewhere in the brain where this particular computation was represented in the activity of single cells. And then, of course, ultimately we found it. Finding it was cool. It was very interesting. But all the failures along the way were also interesting because they circumscribed the space in which you can explore and the region of the brain universe, if you like, in which you can make this discovery. And of course, when we actually did find it, I mean, the work that ultimately uncovered these cells was done in collaboration with one of my oldest friends, was Bill Newsom, who's a well-known scientist at Stanford, who was at the time a postdoc at NIH. And it happened because we sat down at dinner at a meeting. I had given a talk at that meeting about one of our failures. And the failures were striking because in the experiments that fail, the cells respond in a way that's not how you would intuit them to respond. They respond in a way that seems sort of unexpected to somebody who doesn't think carefully about what's in the stimulus. And Bill started out by not believing my results, and that took about an hour. And then after he decided he believed my results, he said, well, the cells in the area that I study, which is MT, wouldn't behave that way. And I said, well, okay, fine. And then from there, that basically became, in effect, a challenge, which we met by, doing experiments and discovering that he was, in fact, correct, that the area, that's at least some of the cells in that area didn't respond in that way. And so that whole process was a mixture and a combination of failure and success, right? If it had been an immediate success, it would have been somehow less rewarding and maybe less distinctive, because to show that something that you predict happens in a particular place but not everywhere, and in a particular way but not in all possible ways, is itself rewarding and satisfying.
So I think that's about all the time we have, Dr. Movshon. So thank you for joining us.
Thank you for inviting me again. And being a recidivist, having done this before, I feel like every 6 or 7 years I should be back here doing it again. I'm expecting an invitation in, should we say 2030?
We're looking forward to it.
Sounds good.
As always, thank you for listening to Max Planck Florida's Neurotransmissions podcast.
If you enjoyed this episode and would like to help support the podcast, please subscribe and tell your friends.